So what are then the components of regulated transcription initiation? Okay, so this is the archetypical situation where you have a polymerase, you have a promoter, and you have a regulator, depending on whether the regulator overlaps with the site of RNA polymerase or is somewhere else. You have positive regulation, negative regulation. We discussed that before. Now, what type of inputs these regulators may receive? Okay, when we say inputs, I mean in material terms, what is that? Okay, so first you may have effectors, and thank you for your question before. So effectors are the chemical inducers or the chemical molecules that will make a regulator to behave in a particular way. So the effectors can be agonists or antagonists. So agonists will be the ones that will change the behavior of the default state of the transcriptional regulator. So for instance, you have a positive an activator, and then you have an agonist, then the activator will in fact go ahead and activate. But it can be also an antagonist. So that means that you may have, for instance, an activator that is always activating, and if you add to the medium one of these antagonists, one antagonist effector, then it will eliminate that effect. Okay? Again, another level of one serious yes-no binary situations. The effectors can be, and this is important, endogenous or exogenous. Endogenous means that, for instance, you may have metabolites that can regulate intracellularly the behavior of given transcriptional factors. Or they can be exogenous, is something that you add from the outside. So if you are used to play with biotechnological projects, the most famous effectors are the exogenous effectors. So you add IPTG, or you add this or that, or you add tetracycline, or whatever. But keep in mind that the vast majority of the transcriptional factors respond to endogenous effectors, not to exogenous effectors. Most cases where you have exogenous effectors are artificial, not all of them, but in many cases they are artificial. We have created them. But the inputs can also be different of chemical molecules. They can be physicochemical signals, for instance. The most, say, known and well-studied physicochemical signal is temperature. So there are many promoters that respond to temperature. Well, in fact, all promoters, to some extent, respond to temperature. But there are some that are specifically, say, programmed to respond to thermal stress. And there is one specific sigma factor that is the one that takes over the entire transcriptional machinery when cells face high temperatures. But there are other promoters that respond to cold temperatures, that respond to desiccation, that respond to oxygen, that respond to nitrogen content, that respond to, you have a bunch of different environmental conditions that some transcriptional factors can interpret as inputs to modulate their behavior. And finally, and this is something very interesting for synthetic biology, say, constructs, is this phenomenon or this signal that is called quorum sensing. Again, this is something that I urge you to pay attention because it will be very important for the rest of the course. So one type of regulatory systems depend on what is called a two-component regulatory system. So when we say activators, sometimes the activator by itself is just one protein. All you need is one protein. You have a repressor, you have an activator that responds to an effector, and that's it. However, there are cases where the signal is not transmitted immediately to the transcriptional factor. It's transmitted with an intermediate, that is a sensor, and then the sensor will in turn interact with the activator and will activate or not transcription of the corresponding gene. So this is a special type of transcriptional regulation that happens in many, many environmental bacteria, not so many in E. coli. Well, you know, if you are not a microbiologist and you use E. coli just as a pet for production of heterologous proteins, coli is, the ultimate microbe. Well, it is not. E. coli is very limited in many respects. And one of the limitations is that the environments where E. coli can live are very, very limited. It can go in the gut and maybe in some water, in some fresh water in the environment. But it cannot go into extreme environments. It cannot degrade unusual molecules. So it has some limitations. However, there are many bacteria out there that can do much, much better than E. coli in different settings because the regulatory networks that they have are way more complex than the ones you find in E. coli. And one component of this regulatory complexity is the abundance of these two-component regulatory systems. There are many, many in environmental bacteria. And for different reasons, what happens is that you have a sensor protein that recognizes the input, and then the sensor protein is a kinase that will phosphorylate or not a regulatory protein, that is the one that really controls transcription, that is active or inactive depending on the phosphorylation state. So this is a special type of regulatory, say, a trick that many bacteria use. But here comes the thing that for synthetic biology purposes is more kind of interesting, and is these quorum sensing systems. So quorum sensing, as I mentioned on passing yesterday, is this phenomenon that has been known now for quite a while, and is a mechanism through which natural bacteria identify whether they are alone or whether they have some friends around. So, and the mechanism that is behind doing that is secreting into the medium a compound that is called an auto-inducer, okay? That diffuses freely out of the cells. So what happens is that if you are by yourself or you have very few friends around, then you will never accumulate high amounts of this auto-inducer in the medium because it will just diffuse out. However, you have a bunch of bacteria around you, and you have what is called a quorum, I mean a limited number, a fixed number of, a threshold of population that is together, then you accumulate enough of this auto-inducer in such a way that the whole community will trigger a simultaneous response, because the same bacteria that produces the auto-inducer has transcriptional regulators that will respond to the auto-inducer beyond a certain minimal level, okay? So bacteria cannot see whether they are surrounded by friends. They just emit signals there. If the signal diffuses out, they know they are by themselves. If the signal increases, they know that they are together with others. And therefore, it's the moment to start doing something together. Again, that's something that looks to me quite nice. So these auto-inducers have been studied for a long time. We know many structures here. And, well, this is one that is super known. There is this homoserine lactones. They are produced by many different bacteria. And so, as a matter of fact, production of these auto-inducers in their natural context is a virulence factor. So many virulent bacteria use these systems. They are enough to start becoming really nasty to the host, or whether they are so little that they better keep a low profile. So there's this bacterium called Pseudomonas aeruginosa that I'm sure that all of you at some point have suffered an infection in the ears or somewhere that use this precise mechanism to kind of develop their virulence program. So if you just have just one or two accidental acquisitions of Pseudomonas aeruginosa, you are fine. But the moment you get a serious a number of cells, then cells know they have enough strength in the numbers to start becoming pathogenic. So that's something that, again, says something about how bacteria evolve all these interesting tricks. So these homoserine lactones are very common in gram-negative bacteria. In gram-positive bacteria, there are also systems of quorum sensing, but they are mediated by peptides, not by these chemical structures. Okay, so basically all branches of the bacterial world have the possibility to use these quorum sensing systems to make these collective decisions about expressing or not a single gene. Okay, so how do we convert all this narrative into engineering, into something that can be subject to some serious engineering? Okay, so then what we can do, given that we have an input and we have an output, we can, one of the things that we can do is to make a transfer function between the inputs and the output. And you have two scenarios depending on whether the promoter or the system is under activation or under repression. So if you are under activation, what happens is that depending on the concentration of the activator that you have, then you have more output, right? So the more regulator that you have in the system, the more active regulator, the more gene expression that you will have. And you can set some parameters like max 2 and K and everything, but typically this will be the behavior of most transcriptionally regulated promoters that by an activator. So when the system, when you don't have activator, the expression is 0. When you have the activator working, then you have a maximum expression. The opposite, logically, is when you have repression. So the promoter, by default, is always working maximally, but then if you repress it, then the expression goes down, okay? So these are the two say, ideal situations. However, note that we have a problem here, and it is that this, say, transition is gradual. You go from here to here, and then you have to go through all these intermediate steps. The shape of the curve may be different in every promoter and everything. And as we discussed yesterday, engineers are not interested in complexity. They want to have simple things. So one big abstraction, I would say a brutal abstraction, that synthetic biologists do normally is to assume that you can represent a kinetic model or convert a kinetic model into a Boolean model. So in the kinetic model, obviously you have a high resolution in terms of the regulation that you observe, but to generate these curves, you need extensive biochemical information. You have to make experiments, you have to pay attention to the reporter, to measure at every point, or to take some points and make an integration curve. I mean, you can do it, but it's complicated, and it's also time-consuming, and you know, we don't have all the time of the world. So in many cases, what has been done, and this is typical of synthetic biology, is to convert these kinetic models with all the details into a simple Boolean model of yes or no. So promoters are on or off. In reality, we know that that does not happen in reality because you know, every promoter goes through many intermediate steps, but for the sake of modeling and the sake of understanding, let's assume that every promoter can have only two states, on and off. And then when they are off, the value is 0. When they are on, the value is 1. And then we can digitalize and convert this kind of gradual transition that we observe in biology into something that is digital. Okay, so this will be the recap of what I just said. So depending on the situation, then you have a repressible promoter and you have an inducible promoter, and each of them can be abstracted, brutally abstracted, into a digital device, okay? So depending on the specific configuration of the promoter and the regulation that we have, depending whether it's positive or negative, or with an inducer, without the inducer, an agonist, an antagonist, etc., we can really develop digital logic gates that, as I explained yesterday, can be implemented. The engineers implement them through electronic devices or electronic components, but in biology, in many cases, can be implemented through transcriptional regulation and transcriptional regulation parts. So this is a slide that I showed you yesterday, just to recap and remember the symbols here. You have an amplifier, you have a NOT gate or an inverter, and you have an AND gate. And as I told you yesterday, and maybe you remember, so you can implement these logic gates with different configurations of RNA polymerase, promoters, and whether repressors or activators, and inducible activators here in this case. But you can make many other logic gates. And well, I ask you also to pay attention to the symbology. You can have an OR gate, for instance. OR gate means that regardless of the input that you have, then at the end it's enough that you have one input, one positive input to have at the end a positive input of the whole thing. So let's imagine, for instance, that you have a polymerase that requires the activity or the action of either one transcriptional activator or another transcriptional activator. So in both cases, the transcription will occur, and therefore you have a typical OR gate that is symbolized with this little, say, half moon here, that is different from the AND that is this straight line. Okay, so this is a symbolic. But you can go farther and farther and complex and get complex logic gates. For instance, you can have an AND gate where you have an AND gate that is negated here. Remember this little bubble that we had at the beginning. So in this case, what happens is that for the promoter, for instance, you may have a situation where a promoter is bound by two different repressors. So it's enough that one of the repressors is there to kill the system here, okay? Or it may also happen that A can interact with B, and then you require the two for this to happen. So, I mean, depending on the specific configuration of the promoter and the specific interplay between the different regulators, the logic of the promoter can be completely different. For instance, you may have also this other a complex logic gate. There is an AND N. So this was N and this is AND N. You know? And in this case, for the promoter to work, you need two things. I'm sorry that this is screwing this up. So you need that you don't have the repressor, and then you have the activator. Okay. So in this case, you have to negate this input here, and then to have a positive input here, and at the end you have an AND, say, computation that will give you transcriptional action. Okay. So how can we implement these pathways or these gates in a completely artificial fashion? The gates that I showed you before are gates that we can interpret and we can abstract as gates, but they are natural. They happen. They are promoters that behave like that. However, let's suppose that you are a synthetic biologist and you want to make new AND gates. So what can you do? I'll show you the very, very first example of an engineered, completely artificial AND gate that was produced by someone called Chris Voigt by the time he worked in the laboratory of someone called, no, wait a minute, this is an inverter. Well, let me go back a little bit. So, well, an inverter is very simple because it's a repressor, okay, so it's not a big deal. In fact, this is the very, very same system that I showed you yesterday. So you have a promoter, you have an expression of one repressor under the control of another repressor. And this is a simple logic gate, it's an inverter. But let's go to the AND gate that I told you before, that it was the first time where a complex logic gate was engineered by humans. You know? So in this case, it goes as follows. This is the AND gate, as I mentioned. You know, you need input A and input B simultaneously to give you at the end a positive output here. So 0, 0, 0, 0, 1, 0, 1, 0, 0, 1, 1, 1, and this is the AND gate. Okay, so you can implement that with electric technology, and it has been done for a long time. But how can you handle this situation in a bacterium, for instance? So this is one example. There have been many other examples, but I'll show you one because I think it's particularly funny. Okay, so what these people did was the following. First of all, you have a promoter that drives expression of a GFP protein, of a fluorescent protein, a green protein, just as an output, as a readout of what is going on. But then this promoter is not a promoter that is driven by a normal RNA polymerase, but is driven by a special type of RNA polymerase that comes from a phage called T7 that is a single protein, okay? And this polymerase recognizes a sequence, that is the T7 polymerase promoter, that is different from any other binding site for the standard RNA polymerase. So there is a specificity between the T7 polymerase and the T7 promoter, okay? So no other polymerase will be able to activate this promoter here. Now, what these people did was to make a variant of the T7 polymerase that had 2 stop codons. That means that if the T7 polymerase is expressed, it will not work simply because in the sequence you have 2 stop codons, and therefore it will simply not be translated. So you have most of the sequence is all right, but in 2 strategic locations of the gene you have 2 stop codons in such a way that the T7 polymerase is not produced in an active fashion. And this, say, faulty gene is expressed under the control of an inducible promoter that responds itself to arabinose. Okay, so let's go in the lower part of the gate. So we have arabinose that induces the activity or activates a transcriptional activator that is called RSC, and then this will activate this promoter here, PBAT, that will in turn transcribe expression or will bring about expression of this faulty T7 polymerase variant that cannot be expressed because it has some stop codons in the way. So that means that if you just add arabinose here, nothing will happen because this T7 polymerase will not be produced, and therefore this will not be expressed through the T7 promoter. Now, the other component, the other input, is a different story. Is a suppressor tRNA that will be able to overcome the stop codons that were introduced in the sequence of the T7 polymerase, and therefore it will enable full expression of the T7 polymerase. But for that to happen, it has to be, you have to produce this suppressor tRNA that is known to, say, overcome the stop codons that are produced in some proteins, and therefore it enables the transcriptional expression of this variant of T7 polymerase. And this suppressor tRNA is expressed itself under the control of a promoter that is selectively activated by a transcriptional factor that responds to salicylate, okay? Okay, recapping. In this case, we have arabinose inducing a faulty variant of T7 polymerase that cannot activate, and you have salicylate that is expressing this suppressor here. Now, if you have only arabinose, nothing will happen because this T7 polymerase will not be produced. If you have only salicylate, nothing will happen because sub-F by itself is unable to activate transcription. But if you have the 2 at the same time, the problem of expression of T7 polymerase will be solved by co-expression of this suppressor tRNA, and therefore, bingo, you will have expression of GFP. And this is what happens, okay? So, if you have very little salicylate or no salicylate, nothing happens. You have very little or no arabinose, nothing happens. But you have the 2, then all of a sudden you have expression of VFP. Okay? Well, as you see, this is not a digital thing. This is not 0, 1. I mean, you go again through this kind of, say, intermediate steps, but this is something, you know, this is more than nothing, you know. And in fact, this made headlines because it was the first time that in a completely engineered, in a completely rational way, someone was able to produce a logic AND gate. You may say, well, AND gate is, you know, any computer has thousands and millions of AND gates. Yes, but there, it was never done before in a biological system in a completely designed way by humans. Okay, so I spent some time explaining you about this AND gate because I think it's a good example on how rationally you can really engineer a phenomenon that in general is associated only to electronic devices, but in this case, everything is implemented in a bacterium. Now, by the same token, and I will not give you so much detail, you can have an another type of AND gate. In this case, is a different story, and in this case, what you have is a promoter that depends on sigma 54, and then the same promoter can be or has to be activated by the concourse of two different transcriptional factors that interact with each other. So only when you have a transcriptional factor called R and a transcriptional factor called S, the promoter acquires this architecture with bending DNA and everything, and then you have expression of GMP. And then in this case, what the authors made, and again, this was a very interesting publication, was to have one of the transcriptional factors, one of the components of the transcriptional machinery, and then the control of one regulator, and then the other, and the control of the other regulator, and again, bingo, you have an AND gate, in this case, implemented with different materials, with different parts, but at the end with the same logic as electronic engineers would do. Well, and you can go on and make things more complicated. For instance, you can implement an AND gate that electronically is very kind of straightforward how to do, but you can do it by combination of an AND gate and a NOT gate. And again, this is something that you can see in some of the papers. So in this case, what they did was to combine the output of an AND gate as the input, or use that as the input for a NOT gate here. So the logic of this construction is therefore that of an AND gate. So we have here an AND gate, okay, and then you have the negation, the inverter, the little bubble here, and you see that in this case what we have is an AND gate that is formed with the two regulators that are required to interact with each other and activate the target promoter here, but then the output here, instead of having direct the GFP, is connected to control expression of our repressor that itself controls or represses expression of another repressor. And therefore, you have a NOT gate, and you have a combination of the two. You have the logic of a AND gate, okay? Okay, you can now start playing with gates and gates and gates and gates. And then, every electronic device, I mean, this is nothing but a last correction of logic gates, okay? So a last correction means many, many. Obviously, in bacteria, we're not there yet, but, there's a big difference between having nothing and having something, And so little by little, what synthetic biologists are doing these days is precisely to generate complex and more complex circuits. I will have an entire talk in this course dedicated to the building of genetic circuits, but the basis of genetic circuits are logic gates, okay? And in this case, this logic was implemented through other methods, but it's just to show that you can use all types of tricks that will generate biological equivalents to circuits and to devices and to kind of elements that engineers use for their genetic, for their electronic devices. Okay? Well, you have many of these. I don't want to bore you with that, but in fact, one of the assignments that I will ask you to read during the weekend will be an entire review of the different gates that you can get with different biological paths. Well, anyway. Okay, so, but you may think that this is very complicated, but I'm sorry to tell you that this is just the beginning. I mean, this is nothing compared to what comes after that, okay? So, and remember I told you that it's not only about DNA going to RNA, you have many, many other things that may happen after that. One first issue. In bacteria, genes are not organized in such a way that you have one promoter, one gene. No. In general, bacterial genes are clustered in operons. So an operon is a cluster or an array of different genes that are co-expressed under the control of a single promoter. Okay? So you have a promoter and have gene A, B, C, D, whatever. This is something that is typical of prokaryotes. In eukaryotes, for the most part, you don't have that. In eukaryotes, and I don't understand why, we'll have to ask them. So you have one gene, one promoter, one gene, one promoter, one gene, one promoter. In bacteria, that, as I told you, appear to be smarter than the rest of the biological systems, instead of doing that, they have developed the ability to express various genes under the control of a single promoter. So the region that happens in front of every gene is a region for binding of the ribosome. And because of that, it's called an RBS, ribosomal binding sequence. So a very typical operon will consist of a promoter, regulated or not, negatively, positively, you name it, and then you will have the ribosome binding site of the first gene, stop the ribosome binding site of the second gene, stop the ribosome binding site of the third gene, and so on and so on. And there are some operons that can be 20, 25 genes that, you know, you can have long RNAs and long operons. Okay, so again, one of the typical operons that have been studied and is in every textbook is the lactose operon. And well, the lactose operon is a kind of historical, say, item that has been studied in extreme detail, not only from the point of view of the regulation here that I explained you before. It can be subject to positive regulation and negative regulation, but also by the fact that it consists of an operon where you have 3 enzymes, where you have a structural gene for beta-galactosidase, a structural gene for beta-galactosidase permease, and a structural gene for beta-galactosidase transacetylase. Look at this. I mean, this is, to me, another sign of how smart bacteria are. Okay? So this operon has evolved for consumption of the sugar called lactose. Okay, so you have the regulatory part that is as I said before, is logical. So this is something that is connected to nutrient choice. When you have lactose and you have glucose, then glucose is easier to consume than lactose, and therefore you prefer to eat first one thing and then the other. You know, when you go for having lunch, you first eat the kind of things, and then at the end you eat a sweet, right? So bacteria have this preference as well. So sometimes they are gourmet, biological systems, and when they are exposed to different foods, they say, well, I take first this one, then the other, and then the other. If they are under stress, they will eat anything. But if they have a choice, they will all be very gourmet and will decide to eat one versus the other. So they prefer to have glucose than having lactose. And therefore, the regulation is such that when they have glucose and they have lactose, they will first consume the glucose and then the lactose. And to do that, they use 3 genes, a part of the regulation here. One of them is a gene for consumption of lactose. So it will clip it's called beta-galactosidase, that will just clip the lactose, and then they produce these monosaccharides, and then they will be eaten. But then there's this other gene that is it's a permease. So it's a gene that will enable the capture from the environment of lactose. Okay? And also there's another gene that will control that the intracellular levels of lactose never get too high. Okay? So this is amazing. Like this is like a perfect design. You know, so you have a regulatory trick here, and you have 3 enzymes that one after the other are expressed in such a way that consumption of these substrates are completely optimized. Okay, so that's really a good take-home lesson from engineers, because what we observe, and is observation, is that the same logic that engineers use to optimize their engineered devices, you can observe implemented with different other types of, elements and different types of parts in biological systems, because the logic for solving the problems has to be always the same if the problems are identical. Okay, now how do we organize artificial operons? And this is something very important, because operons occur in a natural fashion, but then in synthetic biology, there's the most typical endeavor, the most typical project is to construct a new operon, a new pathway with new genes. So to do that, you have to consider various parts, various considerations. You have to put together a regulatory, module, and the regulatory module is a device that is composed of various parts here. So one part will be a regulator that responds to an input, and the input can be positive, negative, an inducer, you name it, as I mentioned before. And then this gene has to be expressed through a Schindler-Gard, through a SDR. Well, the ribosomal binding sites are called RBAses, ribosomal binding sites, but very frequently they are called Shine-Dalgarno sequences because they were discovered by two individuals, Shine and Dalgarno, so Shine-Dalgarno sequences. So in the literature, and don't get confused by that, you find these ribosomal binding sites named either as Shine-Dalgarno, SDs, or RBSs, okay? Don't get confused about that. Okay, then you need a promoter here, and then to express the regulatory part, and then this regulator will target a promoter that will give these POPs, these polymerase per second, this transcriptional current that will go into the construct that you have made for your purposes. So that means that you have biosensor module here with all these parts that will will deliver transcriptional activity, and the transcriptional activity will go into this other construct here. So in these constructs, you have for each gene, you have the necessity to have the ribosome binding in front of the gene, and in these cases, you have what is called intergenic regions. So you have an intergenic regions between gene A and gene B, and intergenic regions between gene B and gene C. And if they are separated, if the intergenic regions are big enough, you need in every case to have an additional ribosomal binding site or Shine-Dalgarno sequence, intergenic region, and so on. But there are some cases, and that happens often under natural conditions, that two genes are subject to a phenomenon that is called translational coupling. So translational coupling means that the end of one gene is coupled or is overlapping the beginning of the other gene. So in these cases, the same ribosome will translate simultaneously two genes without the necessity to have a Shine-Dalgarno, okay? So these are all details that come from molecular biology, but that synthetic biologists have taken into their agenda because they are very important assets that you have to construct complex devices, okay? So again, these are, say, terms and, say, words that I invite you to remember, and intergenic regions, ribosomal binding sites, biosensor models, catalytic models, and so on, okay? But this would be like the typical symbio device that we find in every synthetic biology endeavor. Okay, now, one more thing. What is in an mRNA, okay? There are, in an mRNA, you have not only a coding sequence, you have many other little things that are important for the sake of engineering new properties. So first of all, you have the untranslated regions that go upstream of the coding sequence and upstream of the start codon that in most cases, not in all cases, but in most cases start with AUG. That is the codon for methionine. And then before that, you have a ribosomal binding site, this Shine-Dalgarno sequence that I told you, and then you have this untranslated RNA. So this untranslated RNA, in many cases, does nothing. It just happens to be there. But in other cases, this untranslated RNA can acquire conformations that regulate transcription itself because of the situation where it promotes or it inhibits the entrance of the ribosome. And it happens, in some cases, that these structures that RNA forms in the untranslated regions can be recognized by small molecules, okay? So, and you have what is called riboswitches or riboregulators. So in these cases, it happens that what happens in this region that is not translated is very important for the final outcome of gene expression, okay? So, you can now make a list of everything that, is involved in transcriptional and regulation and in gene expression, and you find all these different possibilities. And then at the end of the RNA, you have an untranslated region that in many cases it does not do much, but it is in some cases important for moderating or for tuning RNA stability. So depending on whether you have here one sequence or the other, the rest of the RNA might or might not be more stable. Simply because in the cells there are these other enzymes that are called ribonucleases. And ribonucleases can chop up and eat the RNA in all possible locations. It can go into the 5' end of RNA or the 3' end of RNA or in the middle of it. So it can be an endonuclease or an exonuclease or a 5'. I mean, you have all possibilities, and all these RNases happen to be in the cell. So this is another level of control of the gene expression flow at the level of RNA stability, because depending on whether the RNA is more stable or less stable, then chances of being translated become higher or become lower. Okay, now here comes a very interesting twist in the whole story that we are addressing today, and is the genetic code. Okay, so the genetic code, I do not need to explain you what it is and everything, but there are some weird things. The first thing that gets very weird is that you have many more codons than necessities for amino acids, right? So you have, I mean, combinatorially, you have 64 possible triplets to encode the synthesis of proteins that are composed of 20 amino acids. You have many, many more codons that you would really need, right? So in principle, you have 20 amino acids. It will be sufficient that you have 20 codons, but maybe 2 or 3 more for starting and for finishing translation. But you have an excess of these things. Now, 18 of these amino acids are encoded by more than one synonymous codon. So isn't it a bit messy? I mean, there are, some amino acids have even 4 or 6 different codons just to encode one amino acid. What a waste of information, right? So that's something that is quite remarkable. Okay, so the other angle of this, the other intriguing angle of this situation is that despite the fact that the genetic code is supposed and claimed to be universal, that's not true. There are some bacteria, in particular some bacteria, some, say, microorganisms and also mitochondria that have some specific codons that do not have exactly the same equivalence in bacteria. So that means that chances are that you can manipulate the codon usage in artificial constructs for specific purposes. So in the last few years, well, not in the last few years, just for quite a while, there has been this question on whether we can rewrite the genetic code, and we are getting deep into synthetic biology and deep biological questions. So as you have so many codons, there are 2 questions. Can we eliminate many of the codons and still have something that is a living system? And number 2, can we reassign the meaning of given codons to encode a different thing? The second part is, the second question, in fact, will be easier because you may think that for serine you may have 6 different codons, and you may say, well, maybe it's connected to the fact that depending on the codons, you may have or less RNAs that associate these codons, and then it can be a mechanism to have the protein being translated faster or slower. But ladies and gentlemen, we have 3 stop codons. I mean, why do we want to have 3 stop codons? With 1 would be more than enough, right? So why do you want to have 3? So what some people have been thinking for a long time, say, well, maybe we can just make a bacterium or make a system where we have less codons, or we have, because if we have less codons, then we can reassign the functionality of the remaining codons to do other interesting things. So this story goes back in history, a long time, many years. So for instance, there's this idea of engineering the genetic code in E. coli. There's this other idea that was mostly pioneered by this gentleman called Jason Chin, that is one of the big names in synthetic biology, and is not just to change the codons, but to invent a new coding system that is based not in triplets, but in quadruplets. And he has some papers here, and this is one of them, in which he uses, I mean, I have to say that the cells are very sick, but still they work, in which some genes have been reshaped to have amino acids that are recognized by tRNA, variants that recognize not triplets, but quadruplets, okay? So that would be one thing. But to me, the most interesting is this idea of codon reduction, okay? So codon reduction is that, as I said, you have an excess of codons. For instance, in the case of serine, you have all these 6 codons. I mean, you have a big excess of codons. I say, well, can you please give up 2 codons or 3 codons? And you will be happy. You are rich in codons, so maybe you can live with half of them and still be happy. Okay, so what Jason Chin did was to take the genome of E. coli, and then he took a look to some specific amino acids that happen to have many codons around. For instance, serine. So in serine, you have all these 6 codons that are synonymous. And then what he did was to eliminate 2 of the codons and leave only 4 out of the 6 to have only 4. How he did that, I will tell you in a moment. But the idea was to have a genome in which every single TCG codon and TCA codon had been erased, completely eliminated from the genome. And the same with these stop codons, okay? So you have 3 stop codons. Then what he did is, well, we can spare 1. So we just eliminate, in all cases, TAG. So you make what he called synonymous codon compression, okay? So instead of having all these codons, you have less codons. And then what he did was to organize a synthetic genome with all these features. You may say, well, how he was able to synthesize a genome with all these reduced, say, codon compression and all that. Well, there will be another class on genome editing. I will not give you in advance, but just to let you know that now this is possible. It's not easy, but it's possible to resynthesize large chunks of DNA for the sake of having a new chromosome that has many, many changes. That's 1 mutation or 2 mutations, but to have thousands of mutations. One super interesting aspect. Okay, let me go back. Okay, so what do you think E. coli without codons would be good for? I mean, you can find any interesting activity or any interesting application of that? So apparently, the cells that have been erased of various synonymous codons for serine, and also for 2 out of the 3, for 1 out of the 3 stop codons, the cells are viable, they grow happily, and so on and so on. So what would be the advantage to have a strain of this type? Do you think that is just an exhibition to show how smart Jason is, or you can find some interesting emerging property in a strain of that type? Okay, well, this is not an interrogation, so I will not harass you with this question. But the super interesting aspect of this strain is that now they become resistant to phage attack. Why? Because phages attack bacteria with the standard genetic code, right? So you have a phage that understands the standard genetic code and it infects a bacterium that has a different genetic code, there's no way the bacteria is infected, okay? And this is what he did. So in this case, we have a phage here trying to infect this bacterium, where he has eliminated 2 codons of serine, okay? So what happens? That then the phage happily injects its DNA here, but when the DNA hits the ribosome, and then one of the triplets that have been eliminated go through the ribosome, then the ribosome does not understand what is going on, and therefore the ribosome will be stalled, and this DNA will not be translated, okay? So in this case, and all these lines here are the different changes that were made in the genome, okay? So it's not like 1 change or 2 or 3, it's thousands of different changes, okay? So the funny thing here is that now we have a strain that is completely resistant to phage infection. And you can imagine the applications that has. You can put this, I don't know whether you are familiar with this problem, but sometimes you have a bacterium that produces a super valuable compound, and you put it in a fermenter, and then all of a sudden it gets infected by a phage, and the whole fermentation goes to hell. And that happens very, very frequently. So you have a robust container of recombinant genes where it is protected against phage action, that's really fantastic. That's something that is really very, very good. So this has been a super big contribution by this laboratory. However, you can say, well, is this, can you see a situation where having a genome that is not amenable to phage attack can be a problem? We can see in this case the advantage of a genome that is not amenable to phage attack, right, in a fermenter or so. But can you see another situation where things would turn completely wrong if bacteria are not attacked by phages? Okay, again, I'll give you another situation. And in fact, I had myself a long discussion with Jason on this particular problem. Our feces contain a lot of E. coli. Okay, and they go to the bathroom, and the feces are liberated into the environment, and eventually it goes to waste treatment plants in some cases, and in other cases they go straight into the oceans. So I don't know if you have a wonder when you go to some beaches why they are so clear and so transparent. Okay, you may think, well, this water is super clean, it's not contaminated, it's wonderful. But okay, in many cases, what happens is not that the water is not contaminated, is that it's packed with phages. So that means that the moment that bacteria get there, the bacteria get immediately destroyed by the phages. And then the water is clear, right? So if some calculations have been made that if E. coli were not attacked and destroyed by phages, all the oceans will be a kind of mess of dirt and excrements and everything, because simply it will not be recycled into the carbon cycle, okay? So phages that sound like very bad guys because they kill bacteria with very nasty mechanisms, at the end are important actors in the whole cycle of elements in the whole biosphere, okay? So if we don't have a system that ensures that bacteria that go into the oceans are killed by phages, then we have a big problem. So what happens is now this bacterium that is produced in the laboratory and cannot be lysed by phages would be released into the environment. Would that be, in principle, that may create a mess, right? So that would be very problematic. Okay, so that's another side of the problem. However, I have to say, and this is again super cool, that in the last few years, there has been a follow-up of these experiments, and it happens that there are phages out there that have invented tRNAs that can suppress the modifications made by us in such a way that in the phage genome, they encode specific tRNAs that are able to overcome the stalin that are produced by this mechanism through which phages could not make it in this type of bacteria. So you have this constant kind of, I would not say war, but this kind of run for, who gets the battle. First, you have a bacterium that is attacked by phages. Then you can make another version that is not, but then phages evolve to counteract what we have made in this, with this trick. So you can see the kind of fascination that biological systems can bring about when you study this mechanism in detail. Okay, but you can go another screw turn. And another screw turn is about having a DNA not formed with natural bases, but formed with xenobases. So xenobases are, as its name indicates, bases that resemble chemically the natural bases, but they are synthetic, they are not natural. So, so another possibility to create semi-artificial living entities, semi-artificial bacteria, is to have DNA that would recognize or we integrate in the structure, not AGTC as every DNA, but AGTC plus other bases that we introduce in the genome artificially. And there have been, well, there's an entire branch of synthetic biology that is called xenobiology, and the idea is to combine the existing network, the existing mechanisms, the existing, say, devices that are naturally occurring in bacteria, combine that with completely artificial inputs that we introduce them for various purposes. So, well, you have many different possibilities, and different laboratories have proposed over the years alternative structures for DNA or DNA-like polymers that still contain the information and still enable base pairing with the same logic and the same chemical structures as we have under natural conditions, but then, for instance, with a different phosphor-sugar exterior, okay? So, for instance, poly-P-glycol or poly-P-hexose or poly-P-hexose. So, these molecules can be made, is no problem. They can contain information, and in some cases, some laboratories have been able to develop DNA polymerases that have evolved to be able to replicate molecules of this sort, okay? So that means that, well, it's not that we have yet a bacterium that has a completely alternative DNA or a bacterium that has a completely alternative codon usage, or, I mean, there you have, we have still like these different excursions in different possibilities of the gene expression flow, but to me, it's very remarkable that we can get inspiration of what nature has already in place to make variants with the possibility that perhaps they work well. So what would be the added value of having, for instance, bacteria or biological, say, things, biological elements, biological agents with alternative genetic codes, with alternative molecules for information storage? Okay, so one of them that would be quite interesting, but still would be in a very, very incipient state, could be the possibility of having genetic firewalls. Because in some cases, and this is one of the discussions we'll have by the end of this course, you know, people or the general public is very reluctant to enable genetic engineers to release things into the environment, and therefore we want to have a degree of containment. So one interesting containment could be that the language and the code for keeping information of the elements or the agents that we release into the environment has been engineered, has been devised in such a way that they cannot exchange information with the neighbors. So if we have a bacterium that has an alternative DNA form or an alternative code or an alternative chemistry, then when it goes into the environment, it will not mix with the others, and therefore it will be a kind of genetic firewall. So if you are curious, there's this paper here that I wrote together with Marcus Smith some time ago, where we will review many of the things that have been done so far to manage the interplay between natural and engineered biology, considering all these possibilities of reshaping very basic functions in the cell, such as information storage. Okay, so I think we're going to stop here and go for a coffee, a little longer break here. I see the coffee over there, so we'll have to wait. And then we'll return to some further issues in connection to the gene expression flow that I hope will be interesting for you as well. Okay, so let's take 20 minutes. Will be enough for a coffee break? The coffee's out there.